Good morning, everyone. Thank you for having me. Thank you to Stefania and Glady that let me do this earlier because I have a little schedule problem. It won't take me too much time. I want to talk to you about target pathogens, the tool that we have been developing in this last year in the University of Buenos Aires, in the Institute of Calculus. Well, first I want to talk a little bit about the past. The first thing is to say that the number of species, bacterial species, have been growing. We have classified more than almost 600,000 right now, and if you go back to 2005, we only got 30,000, and on top of that, the bacteria that we already know are causing us problem because the drug resistance has caused almost 5 million deaths just in 2019, and also the pharma isn't doing very great because the number of new drugs developed and that reach to the market has dropped since 1984, and just right now is getting a little better. So our goal is to develop a sophisticated bioinformatics tool that can simplify the user experience for non-bioinformatic experts, identifying potential drug target efficiently, and establishing a robust and scalable database. So I want to talk a little bit about the pipeline and about our solution. The only thing that target pathogen needs to run is the genome in GBK format. And from that, we got developed an automated pipeline from genome annotation that yields all the proteins from that genome. And from there, we run several protocols, several tools, searching in different databases. We search almost 20 databases to populate our own, essentially searching if the gene is essential, if the gene has any off target in the human proteome or in the human microbiome proteome. And also we do an automated metabolic pathway reconstruction. On top of that, the platform also runs other tools like psort that serves to search for the cellular localization of the protein. And the last module, all of this, except from psort, was present in the first version of the target pathogen that has been running for almost 5-7 years, almost 10 years. But the novel thing that we wanted to do in this new version was to use AlphaFold to generate the structurome of that bacteria. So from there, we also run pocket detection tools too, fpocket and p2rank. So just so you can say how does it look, here's an example of a leucine ligase. This is a picture of a certain part of the protein. Here in blue, we can see the best prediction by p2rank. p2rank does a surface prediction, and fpocket does a volume prediction. So from here, what we want to get is the spots that the protein has to bind some possible inhibitor. So from there, what we got is the pocket prediction, and also we search in the Campbell and the PDB-bind database to get the known ligands for the proteins of the genome. And this is a little picture of how it's visualized in the website. I will be talking about the website in a minute, but from here you got the structure, the name, and some links so you can do a little research on those compounds. And also we want that so we can validate the results from the pocket prediction analysis. So we can see that one of the the binders that I just showed you is actually binding to the predicted site. So from there we got the pocket prediction and the non-ligands for the genome. Just to do a little recap, if we take all the work that we have been doing, we have analyzed more than 30 different species in bacteria, including tuberculosis, Pseudomonas, and Klebsiella. We publish more than 10 scientific papers analyzing each of these bacteria. And here we got a little picture of the target selection page of the website. In the first column, we got the name, the second, the description. Sorry, the first has the locus tag, that is the main key for the database. In the second, we got the description. The third, we got the name. And the last two, I want to start from the last one, is the drugability property calculated by the surface prediction programs. Here we can see that we have done a little filtering. That's one of the features of the page that lets you filter based on the different conditions that you have, that the platform has calculated. Also, you can input your own data. For example, if you got some, I don't know, RNA-seq result, you can input to the web page and use it as a filter and also as a scoring, because the main feature of target pathogen is that enables you to set your own scoring function based on the properties and let you rank all the protein from the genome from the most promising to the less. Here we got that we are only using drugability, but you can also use, for example, localization, and if it has a hit in the essential gene database. So we can get that the first one, the single PTS2, has a score of 7 because it has a high drugability. It's located in the cytoplasmatic membrane, and it's an essential gene. So it makes it a promising target to kill the bacteria. The technologies that we are using are Postgres, a relational database, and Django as a framework for web development. We also spend a lot of effort Dockerizing all the different apps, all the different modules, so you can install it easily in any machine. If you are not using Docker, I highly recommend it to you. And also, thinking on the scalability of the project, we are using Parls, that is a parallelizer for Python that lets you use the different cores of your CPU and lets you scale the project in the moment that you get more infrastructure. Yeah, and it's very easily. So this is what we got so far, and what we want to do in the future, probably in the next semester, we want to use the information from the database, the pocket prediction, and the known ligands to use it as features to develop an AI model that can predict the novel ligands from that information. So, in summary, target pathogen is capable of, it enables a sophisticated bioinformatic analysis without requiring technical expertise from the user. It ensures the reproducibility of analysis across experiments because the pipeline is the same for all the bacteria. It streamlines laboratory processes by filtering out less promising targets, saving valuable time because every reactant in the lab could be really expensive. And it established a robust database to train AI models for novel drug discovery. Thank you very much.